Okay, hello. My name is Nick, and I'm going to cover exercise 3, seedless plants, with you guys. We've got 5 main groups that we're going to talk about. The 3 of them fall in the group of the bryophytes, including mosses, Bryophyta, hornworts, Anthocerophyta, and liverworts, Hepatocophyta. And then we'll cover club mosses, Lycophyta, and ferns, Pteridophyta, a little later on. Just to cover learning objectives for this group of this exercise, um, moving on from that background information in the introduction to plants, we're going to get into the distinguishing characteristics of these different groups and their morphology and how that plays into their life cycle. And uh, this all ties into this transition from water to land and the various adaptations to help make that possible for plants. So bryophytes are non-vascular seedless land plants. They're typically pretty small and found near water because they're highly water dependent. Um, the evolved forms that we are familiar with are the liverworts, mosses, and hornworts. And in all these groups, the gametophyte is the dominant stage of the life cycle, meaning it is larger and longer lived. So these plants may possess stomata or pores that open and close, and this allows for gas exchange but can also help reduce water loss. And you mainly find the stomata on the sporophyte generation, and this is just indicative of that sporophyte generation being less dependent on water, which we'll see play into the alternation of generations as we discuss more plant life cycles. You can also find the beginning of a cuticle, which helps reduce water loss on the surface of the epidermis of these plants. Bryophytes have no true vascular tissue. Like I said, they're pretty small and need to be close to water. They don't have true roots, but instead have these rhizoids, which anchor them to the substrate, found on the gametophyte generation only, which is the dominant generation. The plants lack water transport cells within the tissues or a xylem, and uh, typically tissues are only a few cells thick, which helps facilitate gas exchange and water absorption. Um, and also a thin film of water is required for the sperm of bryophytes to swim from antheridia, or the male reproductive structures, to archegonia, or the female reproductive structures, in order to accomplish fertilization. And all these are reasons why bryophytes are highly dependent on water and moist conditions. Starting with phylum Hepatocophyta. This is the liverwort group, and liverworts are typically flattened, branching leaf-like plants, and this gametophyte stage is called the thallus. They can reproduce both sexually and asexually, and the archegoniophore and antheridiophore are the sexual structures, and gemicups are the asexual structures used for reproduction. Strong evidence suggests liverworts evolved before mosses, and maybe they had diverged even earlier in their evolutionary histories. They are simple plants with a heavy reliance on water, and the sporophyte phase is physically dependent on the gametophyte tissues. So on this slide, you can see multiple images of the asexual structures for reproduction on liverworts. So the thallus is the large leaf-like body of the plant, and then there are these cups that are almost like baskets containing gemi, and the cup is called a gemi cup. Basically, when it rains, water can splash the gemi out, and if they land in a suitable area, they can grow into a new thallus. And again, liverworts also have sexual reproduction, which is accomplished through the use of these structures known as the archegoniophore and the antheridiophore, which are the female and male stalk-like structure growing out of the thallus. And on top of the archegoniophore and antheridiophore are the archegonial head or antheridial head. And again, and just to reiterate, within the archegonial head, you'll find archegonium containing eggs, and within the antheridial head, you'll find antheridium containing sperm. The archegoni archegoniophore and archegonial head is this palm tree looking structure, and the antheridiophore and antheridial head looks more like an umbrella shape. So if we look at the life cycle of Hepatocophyta, the liverworts, and start with the spores, we're starting at a haploid single cell, and these are released into the environment, and under appropriate conditions, they'll form a small flattened thallus, um, and this thallus can form male, female, or sometimes both uh, gamete, um, gametophyte gametangia, or the reproductive structures. Um, it grows and produces these gametangia via mitosis and remains haploid. Um, Antheridiophores are produced for male strains, and archegoniophores are produced for female strains. The antheridiophores house the antheridia, and archegoniophores house the archegonia. The antheridia produce sperm, which will use their flagella to swim and need water to swim through to meet an egg in the archegonium. And it is within the archegonium that fertilization occurs. Uh, this fertilization results in a single cell diploid zygote and is the beginning of the sporophyte generation. This zygote undergoes many cycles of mitosis, and multicellular embryo forms within the archegonium, which is a protective structure underneath the archegoniophore leaves, the palm tree looking structure. And the diploid embryo continues to grow into a sporophyte capsule, which grows directly out of the gametophyte tissue. The capsule elongates, and spore mother cells, which are diploid within the capsule, undergo meiosis to form multiple haploid spores, which burst out of the capsule and are released into the environment through the wind. And if a spore lands in a suitable habitat, it will germinate and grow, beginning the gametophyte generation, the thallus, and completing the liverwort life cycle. And again, liverworts can also reproduce asexually using gemicups, which form on the thallus surface through mitosis and are haploid. And each cup houses gemi, which is a cell or collection of cells that can detach when water splashes them out of the gemicups, and the gemi are ejected into the environment. The gemi will grow into a new thallus under suitable conditions. And now I'm going to switch to the whiteboard and draw out the life cycle for you guys. And we're going to start here with spores, which makes sense, right? So first off, we've got our spores, which are haploid. And under the right conditions, spores will go through mitosis to to form a thallus. So we said the thallus of the liverwort is kind of this lobed leaf-like shape, and this is a um, multicellular haploid. The thallus will also undergo mitosis to produce the gametangia, or the sexual reproductive structures, which can come in the form of archegonia, which are kind of palm tree shaped, or an archegoniophore, I should say, or they can come in the form of antherydiophore, which are kind of these umbrella shaped structures. So if we look more closely at this, though, the if we zoom in on the head of these archegoniophore and antherydiophore stalks, known as the archegonial head or antheridial head, we'll be able to see the actual antheridium and archegonium. So I'm drawing the stalk down over here, and starting with the archegoniophore, the archegonium are basically going to just be these pockets in each frond, let's say, of the palm tree, and the antheridophore is going to have them on, instead of underneath, on top, so that water can splash sperm off of this structure and then hopefully onto a archegonium, so sperm can fertilize the egg cell. Um, And something that might help you remember, archegonial or archegonium. There's the word egg is kind of in it, so you can associate that with the female structure. And then antheridium, the anther kind of sounds like antler, and that's something that like a male deer would have. So just something to consider. But if we continue on with our life cycle here, our sperm and egg, our gametes, are going to come together through fertilization. Um, and fertilization is going to occur in the antheridium, or excuse me, in the archegonium. Um, this is important to know because the tissue is actually the maternal tissue is actually protecting and nourishing the zygote, which is the sporophyte generation. Okay. Um, so this again is here's our palm tree structure, and I'm just going to show. So first we've got a zygote here. Um, this would be diploid, single cell, which goes through mitosis to uh, form the multicellular sporophyte embryo. Remember that this is the start of the sporophyte generation. And it's diploid. And this is going to keep growing through mitosis until the spore mother cells go through a stage of meiosis, and meiosis is going to reduce our ploidy to um, 1 or 2 haploid. And again, that is meiosis that is reducing the ploidy for these spore cells. The spore cells will burst out of the spore capsule, and if they land in a suitable location, form, grow into the new thallus, the new gametophyte. This is the completion of the life cycle, but we can't forget about the asexual life cycle, which is basically a thallus and a cup-like structure on the surface that houses multiple gemi, which are a collection of cells that can be splashed out So if it's raining, uh, and the water splashes out, um, it's possible that some of these gemi will also splash out. And if they land in a suitable location, they will grow in through mitosis into a new thallus. This is all occurring in the gametophyte stage, so it is always haploid. And this thallus can go on to produce sexual or asexual structures for reproduction in the future. Hopefully this isn't too confusing, and I appreciate you watching.